Okay. Rightio, do you want to flick over to my picture? Yeah. Hello, folks. I'm facilitating this coffee seminar today. I'm an assistant professor at in the Centre of Hort Science and work on mostly on macadamia. That's what I'm standing next to there. And I'm the coordinator of the science seminar series. Can we have the next slide, Michael? So I'd like to start by acknowledging country. The University of Queensland acknowledges the traditional owners and their custodianship of the lands on which we meet today. We pay our respects to their ancestors and descendants who continue cultural and spiritual connections to country, and we recognise their valuable contributions to Australian and global society. Can you have the next slide, mate? So I just wanted a couple of housekeepings for this. First of all, I just want to say that we're just in a bit of a changeover period with Coffee Comms, and they're doing a great job catching up on top of presenting the seminar series. And hopefully the last seminar last week wasn't too bad with no glitches. Hopefully today will be fine, but just a bit of understanding there. So the seminar is going to go to 1 o'clock. You'll note for question and answers, there's a Q&A tab at the bottom, so not the chat button. So if you've got any questions throughout the seminar or at the end, you can type your questions in there, and part of my role is to find the tricky ones for Michael to answer. And we'll do that at the end of the session. So Michael, can you just go to the next one? So I just want to introduce Michael. So he's joined Coffee over the last 12 months. He received his PhD Michael Uvadi has joined as Professor of Legume Genomics at Coffee. He received his PhD in plant biochemistry from ANU in 1989. His primary interests are symbiotic nitrogen fixation in legumes and the use of genomics to advance legume breeding. He was elected Fellow of the American Association of the Advancement of Science in 2012 for his work on symbiotic nitrogen fixation. So Michael, I'll hand it over to you and I'll see you at the end.
Thank you very much, Craig, and hello to everyone. Good afternoon. It's my pleasure to present today. I've got a little bit of work in the background outside my office, and hopefully that's not going to disturb folks. But what I'd like to do today is review my work in nitrogen fixation research over the last 20 years, and then spend a part of the time thinking about and discussing what I'm doing here at Coffee at UQ. So to begin, I should say that legumes are a crucial source of protein and other nutrients for humans and animals, and a sustainable source of nitrogen for agriculture. And that's what I'm talking about today, symbiotic nitrogen fixation. Nitrogen is typically limiting in most ecosystems and in agricultural systems, as you can see in this slide. So what we have in the first panel is a meta-analysis of several hundred studies of terrestrial, freshwater, or marine environments where the biomass produced in those systems was monitored with or without the addition of nitrogen, phosphorus, or both. And this is a log ratio, so anything above 0 is an increase. So adding nitrogen in these systems typically leads to an increase in productivity, as does phosphorus, and adding the two together tends to have synergistic effects on productivity. We see that also in agricultural systems, as you can see on the right-hand panel, where we're looking at the effects of adding nitrogen to maize production. So without nitrogen, grain yield is about 4 tons per hectare, and with nitrogen, virtually double. And you can also see the effects of adding water in these systems as well. So again, maximizing the use of nitrogen in agricultural systems requires not only nitrogen, but water and phosphorus and other limiting nutrients. Over the last 60 years or so, recognizing the fact that agricultural systems are typically nitrogen limited, led to the use of nitrogen fertilizers in the 20th century from about 1950 onwards. using nitrogen produced through the Haber-Bosch process. So over the past 60 years or so, our use of nitrogen has increased from about 10 million tonnes of nitrogen per year to well over 100 million tonnes today, as you can see in that left-hand panel. At the same time, the world population has increased from just over 3 billion to well over 7 billion. So this tenfold increase in nitrogen was a driver that enabled population growth, a doubling of population growth in my lifetime since 1961. That also enabled increases in the relative production of crops and animals, as you can see in the right. So cereal crops, for instance, have increased, the production of cereal crops has increased about threefold in this period. It's been argued that without fertilizer nitrogen, there'd be some 2 to 3 billion fewer people on the planet today. So fertilizer nitrogen has become crucial for agriculture as we know it. In fact, our use of nitrogen fertilizer has doubled the flux of nitrogen through the global nitrogen cycle. So you can see that in the brown arrows here, the Haber-Bosch process, nitrogen fixed through cultivated legumes and through and burning of fossil fuels, and the oxides of nitrogen that are produced there have virtually doubled the inflows of nitrogen into the terrestrial ecosystem. Ironically, despite this huge influx of industrial nitrogen in the globe, there are parts of the globe, as you can see on the map on the right-hand side, that still have insufficient nitrogen for decent yields. So we've got this kind of dual problem of nitrogen now. We've got too much nitrogen in parts of the world and too little in other parts. Our interference in the global nitrogen cycle is beyond what is considered a safe operating limit for humanity, as you can see in the left panel here. So alongside biodiversity loss and climate change, our interference in the nitrogen cycle is one of three activities that are really unsustainable in terms of life as we know it. On the right-hand panel here, you can see major sources of nitrogen, anthropogenic nitrogen, that are going into the global nitrogen cycle. And you can see that agriculture is a major source of ammonia pollution, nitrous oxide pollution, and dissolved nitrogen pollution going into the environment. While burning of fossil fuels in our cars, for instance, is a major source of other oxides of nitrogen. In Europe, they've paid attention and tried to quantify the impacts of reactive nitrogen in the environment. And as you can see here, the estimated costs are run in the order of 100 billion euro a year just in Europe, with effects on climate, ecosystem health, and human health. And agriculture, as you can see, has a major impact, more of the order of tens of billions of dollars. So many-fold more than the actual cost of nitrogen production are the costs to human health and ecosystem health. Legume nitrogen fixation is a way of reducing the reactive nitrogen that is lost from agricultural systems. And this slide shows the biochemistry of nitrogen fixation in legumes. So it's a reaction that's carried out by bacteria that reside in legume nodules, and they possess a complex called nitrogenase that carries out this reaction, the reduction of nitrogen to ammonia, at the expense of a large amount of energy in the form of ATP and reductant. In essence, this symbiosis in legumes is an exchange of reduced carbon derived from photosynthesis in the leaves, which is transported to the nodule, provided to the bacteria, and runs a nitrogen fixation process. So it's an exchange of this reduced carbon for reduced nitrogen in the form of ammonia. This process injects about 50 million tonnes of nitrogen into agriculture each year, which is a little less than half of the nitrogen that's provided by fertilizer nitrogen from the Haber-Bosch process. The value of the biological process in legumes is about $50 billion a year globally. This slide was taken from a review by Zhang et al. Let's see if you can see here, and it compares the nitrogen inputs into various agricultural systems in this column here, and where you can see that cereals consume more than half of the nitrogen fertilizer applied to agriculture. These inputs, I should say, include fertilizer nitrogen as well as nitrogen fixation and nitrogen that's also deposited from the atmosphere, which is a smaller amount, but the inputs consider all of those inputs. The column on harvested end shows how much of that nitrogen is harvested in the grain and other products that are removed with the harvest. So a total of 74 million tons of nitrogen per year is harvested of the 174 million that enter the agricultural systems. That leaves a surplus of about 100 million tons of nitrogen a year that's in the agricultural systems and available to leakage. So that's where a lot of the pollution that I mentioned comes from, this surplus nitrogen. It's interesting to look at the nitrogen efficiency in the different systems. In cereals, it's typically around 40% of the nitrogen that's inputted into the system is harvested. So 60% is vulnerable to losses. In contrast, in legumes such as soybean, there's a nitrogen efficiency of 80%. So very relatively little of the nitrogen enters these systems, primarily through nitrogen fixation, is vulnerable to loss. And that's one of the reasons legumes are so good in terms of nitrogen pollution or lack thereof. So over the last 20 years at the Nobel Foundation and before that at the Max Planck Institute of Molecular Plant Physiology, I've been using genetics and genomics to explore the genetics of nitrogen fixation in model legumes. And the sort of kind of the mantra, I suppose, over the last 20 years is that, through functional genomic studies of model systems, we would gain knowledge of gene function that would ultimately lead to crop improvement. So for the work that I'm going to describe today, we focus primarily on Metacargo truncatula, which is a model legume. It's a minor forage species. It's used, for instance, in South Australia as an annual forage species. It's related to alfalfa, which is a perennial forage, which has a more complex tetraploid genome. Truncatula, Metacargo truncatula, on the other hand, is a diploid genome, relatively small, at 550 megabase pairs. It's self-fertile with good seed set, so it's good for genetics. It's transformable by both Agrobacterium tumefaciens and rhizogenies. There are multiple mutant resources available for genetics, including EMS, transposon insertion mutant populations, deletion populations. More recently, in addition to a reference genome, sequence from close to 300 additional genomes has been obtained, which is enabling genome-wide association studies, and I'll talk a little bit about that later. And also early on, we developed in my group the gene expression atlas for Metacargo truncatula, which enabled us to identify, for instance, genes that are induced during nodule development as targets for some of our genetic work. One of the important genetic tools that was developed at the Nobel Foundation for the research community was this transposon insertion mutant population, called a TNT1 population. It took a retrotransposon from tobacco and introduced it into Metacargo through transformation with a starter line that had 5 copies of the TNT. That TNT can be mobilized through tissue culture, through somatic embryogenesis. And because it's a retrotransposon, it produces mRNA from the initial DNA insertion in the chromosome. That mRNA is reverse transcribed into double-stranded cDNA, which then inserts at random in the genome. So from those initial 5 starting points, we have approximately 1 million insertions in the Metacargo genome in about 20,000 lines, which is summarized here in this slide. So with those starter lines, we regenerated about 22,000 lines. We've taken DNA from those lines using tail and inverse PCR and sequencing. We've been able to sequence the flanking sites adjacent to insertions in those 20+ plus thousand lines with about 350,000 flanking sequence tags. So that information is in a database. And so folks that are interested in identifying insertion mutants in any gene of interest can go to that database, discover lines with insertions, and then order those lines and study the effects of those insertions on gene function. In addition, DNA from those lines was pooled and used initially in PCR screens to identify insertions in over 1,000 genes, and about with a close to 90% success rate. So for any gene of interest, if we did a PCR screen on this DNA, we're able to recover insertions for about 90% of those genes. So this is a tool for reverse genetics, where you start with a sequence of interest and then work towards a mutant and then characterize a mutant. The population has also been used for forward genetics, where phenotypes of the mutant population have been screened, and then the genes affected have been identified by looking for insertions in specific genes within those mutants. So we've screened that mutant population together with collaborators, and we've identified over 100 mutants affected in nodulation or nitrogen fixation. And you can see a panel of some of those mutants here, with panel A showing the typical wild-type nodule, which is often coralloid with multiple meristematic foci in the nodules. These are also typically pink from the hemoglobin protein, the major plant protein that's expressed in nodules. Many of the mutants are white and non-functional. They don't express hemoglobin, and they're also defective in other aspects, so they don't develop to let's say a functional state. We have mutants that make no nodules at all, and other mutants that are supernodulating mutants. Over 20 years, through genetic studies in Medicago and Lotus, we now have something like over 200 genes that have been discovered that are necessary for nitrogen fixation. And these timelines, if you like, on Medicago and Lotus show the different genetic approaches for mutant populations that have been used, from X-ray mutagenesis and EMS mutagenesis in the 90s through to antisense RNAi approaches at the turn of the century. The TNT mutant population was really brought online in about 2007, and has subsequently led to the discovery of many genes required for nodulation. And more recently, CRISPR has been applied both in Medicago and in Lotus to target genes for mutagenesis and functional studies. So if you go to Roy et al., this is a paper we wrote a couple of years ago now, or published a couple of years ago, that has a nice summary of what we knew at the time in terms of the genetics of nitrogen fixation. I want to go through with a couple of examples from my own lab on genetic characterization of genes. This is a figure taken from our review, and I just want to use it to kind of introduce the developmental process of nodulation. On the slide, you'll also see genes that are involved in hormonal processes that are required for nodulation, but I don't want to go into that detail. Simply want to point out the various steps. The symbiosis begins with chemical signaling between the plant root and bacteria in the soil. Flavonoids from the plant serve as a trigger for induction of so-called nod genes in the bacteria. These produce nod factor, which is a lipocyto-oligosaccharide molecule. It's analogous to chitin from fungi, so fragments of the chitin surface or the fungal surface serve as signals in their own right and lead to, in the case of mycorrhizal fungi, lead to mycorrhizal symbioses. So this bacterial symbiosis is actually using part of that signaling process, and the bacteria are producing signals that were initially recognized by plants to signal the presence of fungi, probably at the time that plants emerged on land 450 million years ago. So these signaling processes are ancient, but the nodulation symbiosis evolved around 60 million years ago. And so it's borrowed some of the machinery and the genetics that was present for 400 million years prior to the evolution of nodulation and nitrogen fixation. In any case, these bacterial signals serve to trigger events in the plant via receptors. Those events lead to infection of the root hair by bacteria and eventually infection of underlying cortical cells. These are events that are actually common with mycorrhizal fungi. So intracellular infection is another shared symbiotic phenomenon, if you like. What's different between nodulation and mycorrhizal symbiosis, however, is the fact that cortical cells divide during nodulation and ultimately lead to the formation of a new organ called the nodule. So that's a unique aspect of this symbiosis. So the bacteria that are infecting cells are eventually infecting cells of an emerging nodule, and those cell divisions form meristems, which in the case of indeterminate nodules remain active, so that the nodule can grow over time and form the sort of coralloid structures that I mentioned before for metacargo. In the case of soybean and the so-called determinate nodulators, that meristem is active only transiently and leads to more spherical structures. So you have these indeterminate nodulators like metacargo and determinate nodulators like soybean. So the first example I want to give, let me just check my time to make sure I'm not going to go over time here, but the first example I want to give is a gene that we called vaporin, which came from a mutant that was able to nodulate barely, and also allow ingress of bacteria into the infection threads, as you can see here. But those infections rarely or never really led to infected nodules. So what you can see for the wild type is a larger nodule becoming pink. The infection thread is well defined. That delivers bacteria. These are GUS expressing bacteria that are stained, so you can see them in single file running down the infection thread towards the cortex. Eventually, those bacteria pass through the epidermal layers into the cortical layers and are deposited in nodules in the wild type case. In the case of the vaporin mutant, we never saw infected nodules. So this figure here shows number of infected nodules in the wild type, uninfected primordia in the wild type, and infection events, these sorts of things in the wild type. What you can see in the mutant is a lot of these infection events and some uninfected nodule primordia, but no infected nodules. So that's the phenotype of these mutants. Interestingly, the vaporin mutant was also defective in mycorrhizal symbiosis. I mentioned this sort of commonality between the two symbioses, at least with respect to infection of the microbe. What you can see with wild type infected with mycorrhizal fungi is a mixture of hyphae, vesicles, and arbuscules. These are the mature symbiotic structures that serve as the nutrient exchange structures in roots between the fungus and the plant cell. So in the wild type, you see lots of arbuscules at these different time points of the symbiosis, 14, 28, and 42 days post-inoculation. In the vaporin mutant, although we see hyphae and vesicles, we never saw arbuscules. So the infection process was also impaired with respect to mycorrhizal accommodation in the vaporin mutant. So this is one of the so-called common symbiotic genes. It's required for both nodulation and for mycorrhizal symbiosis. Now, when we took an intact or a functional version of the vaporin cDNA and expressed it in plants under a 35S promoter, we were able to recover functional nitrogen-fixing nodules and also functional arbuscules in these complemented mutants. So that's demonstrating or confirming that vaporin was the defective gene in that particular mutant. Another example that I want to give of functional genomics was done actually a few years earlier in Germany when we were working on the other model legume system, Lotus japonicus. here we used RNA interference to study the function of 3 hemoglobin genes. Now, I mentioned earlier, these are the major or the predominant plant protein in nodules. They give it the pink color you see here, so it's analogous to myoglobin in human cells. It's an oxygen binding protein. It keeps oxygen levels low while moving oxygen around rapidly in infected cells. So when we used RNA interference, we were able to drop the expression of the 3 nodule-specific hemoglobin genes, LB1, 2, and 3, down to about 1 to 2% of their normal wild-type levels. So this is a log scale here, without affecting the expression of 2 other hemoglobins, the so-called non-symbiotic globins 1 and 2. So the RNAi was fairly specific to these related leg hemoglobin genes, the so-called nodulin genes. You can see in a Western blot that compared to the wild type, where you get high levels of hemoglobin expressed, the mutants or the RNAi lines that were generated had no hemoglobin expression, leg hemoglobin expression. And nor did these nodules express bacterial MIF-D. This is one of the bacterial subunits of nitrogenase. So while the wild type plants enabled the bacteria to differentiate into a nitrogen fixing form, that wasn't the case in our RNAi lines. So you can see the gross phenotypes of these RNAi lines compared to wild type. So under nitrogen fixing conditions, the wild type grew well, but these RNAi lines, the hemoglobin RNAi lines, grew very poorly. They were unable to fix nitrogen. On the other hand, when those plants are presented mineral nitrogen, those RNAi lines grew just as well as the wild type. So the hemoglobin genes have a specific role in nodulation and nitrogen fixation and are not required for normal plant growth and development under non-nitrogen fixing conditions. If you look into the nodules of these RNAi lines compared to the wild type, this is looking at oxygen levels from the surface of a nodule into the center of a nodule. And in the wild type, you can see that for much of the central part of the nodule, oxygen levels are essentially undetectable. So with hemoglobin present in nodules, oxygen levels can be kept at very low levels that are compatible with nitrogenase activity. What I didn't say earlier was that nitrogenase is oxygen labile. So it can only function when oxygen levels are very, very low in the nanomolar range. Typically, oxygen levels in plant cells are in the hundreds of micromolar range. So oxygen levels have to get down to about 5 orders of magnitude less before nitrogenase can function in these nodules. In the RNAi nodules, oxygen levels are quite a bit higher, and in fact are still detectable even in the center of the nodule. So oxygen levels never get to a level that's compatible with nitrogen fixation. So those are 2 examples of the use of genetic approaches to understand gene and protein function in nodules. Over the years, we've published about 16 papers looking at various genes involved in oxygen binding, nutrient transport, infection, etc. And you can see a list of publications there. That's added to the total knowledge of genetics of nitrogen fixation, where we today have knowledge of about 200 genes that are required for symbiotic nitrogen fixation. So it's a complex process, and it's a process we haven't really been able to harness in terms of improvement. So although the promise of molecular biology is to take genomic and genetic information and knowledge of that and use that to improve crops, in the case of nitrogen fixation, that hasn't been the case yet. Now, there are obviously some nice examples of where functional genomics of simple traits like lignin biosynthesis or disease resistance have been translated into crop improvement, but that's not the case for nitrogen fixation, and perhaps never will be if we take a gene by gene approach. Those stumbling blocks for the translation of knowledge and nitrogen fixation, as with other complex traits, is that, first of all, quantitative traits like nitrogen fixation are determined by many genes, and therefore optimizing these complex traits will probably involve many genes, more than we can probably manipulate through transgenics or through genome editing. There's also a lack of acceptance of transgenic plants for foods, which means that the impetus to develop better nitrogen fixing legumes is not there, at least not using genetic and functional genetic tools. Also, the cost of translation is high with transgenic. So with this in mind, we decided to do what breeders typically do, and that is to try to harness natural variation in order to not only understand the genetics and genomics of nitrogen fixation in model legumes, but also to ultimately harness that natural variation in more traditional approaches to breeding. So, as a tool in this work, we've taken advantage of a resource developed by Nevin Young in the USA, where he and collaborators collected nearly 300 wild accessions of metacargo from the Mediterranean basin, which is its site of origin. And they sequenced these genotypes, all of them at least 5x coverage and at least 30 lines of 20x coverage. That led to a discovery of over 1 million SNPs that can be used in genome-wide association studies. Now, we've taken that population and asked the question, what is the variation in that population for nitrogen fixation effectiveness? And we define effectiveness as the ratio of biomass that a particular genotype can produce under nitrogen fixing conditions, which you can see on the x-axis here, divided by the biomass that can be produced when the plants are grown on mineral nitrogen. And what you can see in this figure is that there's a good spread of biomass production from large to small plants. Generally, plants that grow better or grow large on mineral nitrogen also grow large under nitrogen fixing conditions. But if you take any one particular biomass under nitrogen fixing conditions, you can see that there's a range of biomass that's possible under mineral nitrogen conditions. So in other words, there's natural variation for effectiveness defined as this ratio of biomass under nitrogen fixing conditions divided by biomass under mineral nitrogen conditions. And that's also shown in the right-hand panel. So this is effectiveness. We have some genotypes that grow just as well on nitrogen fixing conditions as they do with mineral nitrogen, but the vast majority produce less biomass under nitrogen fixing conditions. You can see it's a quantitative trait, and by virtue of the fact that we have a million SNPs available in this population, we're able to map regions of the genome associated with nitrogen fixation effectiveness. And you can see one of the Manhattan plots here where these peaks represent loci that are contributing to effectiveness. And this particular gene is a molybdenum transporter. Molybdenum is a key cofactor. Molybdate is a key cofactor of nitrogenase. So it's interesting that this particular molybdenum transporter gene is associated with efficiency, presumably because it helps to get molybdate to the bacteria within nodules. But in addition to this, we see a range of interesting candidate genes, many of which haven't been discovered before. So this approach of using natural variation is a rich approach to discover new genes involved in nodulation and nitrogen fixation that may be more subtle and perhaps not easily found in a normal genetic approach. These results also give us hope that the use of natural variation and genomic associations like this could be harnessed in breeding programs in future. So that's kind of how my thinking has evolved over the last years, with the desire to capture natural variation through genomics. And in this current role at Coffee, we're starting to create genomes for target species, and I'll talk about some of those in a moment. And we want to use association genetics and genomic prediction to assist breeders in improvement of not only simple traits, but complex traits like nitrogen fixation. So how are we doing on time? Let me just check my time. I've got probably 5 or 10 more minutes. I want to just race through quickly where we're currently at. So with my new position, the focus is not on the temperate legumes of the past that I've worked on the past, but a focus on tropical legumes that are directly used in agriculture, and they include species like mung bean, soybean, and potentially in future plants like pigeon pea. Now, our breeders for mung bean, for instance, have targeted a number of areas for improvement, either of improvement of yield or yield stability. These include areas of abiotic stress tolerance, physiological constraints, such as indeterminate flowering. Biotic stresses are important to our breeders who are trying to get disease resistance, for instance, in mung bean. And as part of a team at Coffee and collaborators in DAF, we're working to develop this type of integrated approach to predictive breeding, if you like, where we take the natural variation available in gene banks for mung bean, for instance. We use pangenomics in conjunction with high throughput phenotyping to be able to establish links between genetic variation or genomic variation and trait variation, and through genomic prediction and other approaches, combine or select preferred genotypes with associations to traits of interest into progeny. So what we're looking at is shifting from phenotype-based breeding to genotype-based breeding, and I'm doing this work in collaboration with a number of people, especially Millie, Lee, and Meryl, Ryan, and Daff. So to summarize what I've said so far, legume reference genomes and functional genomics have enabled a rapid advance in our understanding of legume biology, especially as it relates to symbiotic nitrogen fixation in model species. Translation of this knowledge to crop improvement has been slow, however, and I believe, and I think my collaborators believe, that pangenomics will enable discovery and more rapid utilization of genetic and genomic diversity for crop improvement in future, not only of nitrogen fixation, but some of the other traits I mentioned on the previous slide. The last 5 minutes, I just want to briefly talk about some of the work that I've been doing with DAF colleagues in the area of forecasting or planning for work in pulse proteins. So you may be aware that CSIRO recently published a CSIRO Futures report on a protein roadmap for Australia that looked at current protein production in Australia, which is worth about $56 billion a year from a variety of sectors, and forecast growth in this protein industry in Australia to either 76 billion or as much as 89 billion, depending on whether or not technology is applied to the growing industry. What they looked at are a number of potential growth areas, amongst which are plant-based protein products, which at the moment have a relatively small share of the market, but have the potential to grow up to about 9 billion by 2030, provided that technology is brought to bear. Amongst the plant-based protein sources, legumes are particularly attractive. In fact, existing plant protein products consist mostly of legume products. About 2/3 of the protein in these manufactured protein products come from legumes, primarily soybean, but other legumes contribute, and wheat-based products represent the remainder. So there's an opportunity as the growing protein industry expands to deploy or to utilize legumes, not only soy, but other legumes. And in fact, in Queensland, those legumes include the list here. These are, well, on an Australian level, these are the legumes that produce protein at the moment. They're mainly exported as whole grains. Within Queensland, Queensland's a major producer of chickpea and of mung bean. The other legumes on this list are cool season legumes, grown mainly in the south. Chickpea is also a temperate legume, but it's grown in winter in Queensland, and as I said, Queensland is the largest producer of chickpea. So there's a variety of legumes that could fit the bill for protein sources in future. And this slide simply shows the breakdown of legumes within Queensland. So we have some of the tropical legumes, pigeon pea, mung bean, soybean, that are grown in summer, as well as the temperate legumes that are grown in winter, chickpea and fava bean included. The production of these is sufficient to meet the expected need of protein in the manufacturing industry. For instance, chickpea would certainly provide the predicted need. Soybean, as you can see, currently is relatively low in terms of production in Queensland, although there's potential to grow soybean, as is the case also for pigeon pea, which isn't grown much at all at present, but has huge potential within Queensland. So there's a number of legumes that could serve as feedstock for protein manufacturing. And as part of the DAF coffee initiative, we hope to work together to expand and diversify pulse protein products with this growing opportunity. I won't go into the details of why Queensland, apart from the fact that we grow legumes at present and there's great scope to increase that. We also have infrastructure in place or being developed to enable processing of any pulse proteins, for instance. So in our discussions with DAF colleagues, we see a number of areas being brought to bear on the pulse protein possibilities, including plant breeding, systems agronomy, food science and technology, and market intelligence, with the aim of improving varieties with proteins tailored for the end users, northern expansion of legumes through systems agronomy, a reduced environmental footprint, so sort of selling the environmental credentials of legumes for sustainable protein production, etc. So overall, we see sort of multiple cogs in this interaction between DAF and coffee that involve plant breeding and systems agronomy to service the existing whole seed markets, but it also interactions with food science and ingredient manufacturers to service the growing demand for formulated foods. So where I'm at in my thinking now, after the various discussions with DAF colleagues and coffee colleagues, is how do we fund these types of interactions, and in particular in the area that I'm interested in, how shall we fund plant breeding? So these are some of the ideas that I'm starting to work with colleagues on. We're looking at submitting an ARC proposal for an industry transformation research program that would focus on predictive breeding of legumes for the tropics. I'm also planning to submit a discovery program grant that focuses more on symbiotic nitrogen fixation. And I'm also interested in discussions of a possible future CRC for tropical legumes in Queensland for sustainable food, forage, fuel and feedstocks. With respect to the transformation or industry transformation hub, the ARC proposal that I'm currently developing with colleagues, we see 4 technology aspects to that, including pangenomics, trait analytics, prediction and testing, so sort of genomic prediction, if you like, and testing, as well as genome editing, as technologies that can cross current and potential future legumes that are grown in Queensland. So we're looking at multiple species being accelerated, if you like, breeding of these being accelerated via these various technologies. So what's transformational about this type of approach? Well, currently breeding of legumes in Queensland is largely based on phenotypic selection in field trials or field-based trait selection, if you like. In future, we see a real opportunity for genomic selection based on the genomic sequence of individuals. Also, I think I see opportunity for novel traits, symbiotic nitrogen fixation, seed structure and composition, especially for the emerging food industries. Comparative physiology, by looking at multiple related legumes, I think we can learn from some, for instance, soybean, what makes soybean so good, and perhaps use physiological comparisons to drive improvements in some of these others. I believe AI-guided, artificial intelligence-guided trait optimization will be important to this process. Genome editing for simple traits at least, and then speed breeding. By growing plants under ideal conditions in growth rooms rather than out in the field, we can aim to have 5 or 6 generations per year instead of 1 or 2 through classical means. So that gives, I think, a little bit of a flavour of where I'm headed in my research program and how I see interacting with others. And so the final slide I just wanted to use to thank my former colleagues at the Nobel Research Institute. Sorry there, I've lost my pointer. My colleagues and collaborators at UQ, and also my collaborators in DAF and QUT. So with that, thank you very much, and I am done. Are you there, Craig?
Yeah, thanks very much. No, excellent, Michael. It's really great. Really enjoyed the overview you gave, both of the functional work and the future work, and hopefully we'll continue to see more of that from senior QUAFI researchers. So there's an opportunity for a couple of minutes of Q&A. So if anybody's got any tough questions for Michael, that'd be great. But I mean, I've I really enjoyed some of the things. A lot of it's not in my area, but some of it is. But there's a couple of things I'd like to start off by asking. I was interested in the where you transition from your functional to your the GWAS type approaches, and particularly picking up some of these genes with the GWAS. And I was interested of the other way of, did you pick up a lot of those functional genes that you'd been investigating, variation for those? Or like, because you mentioned you had a lot of new genes, but yeah.
Yeah, a few. I think the molybdate transporter that I showed was an example of one of the genes that had been discovered through the forward genetic or approaches, or actually, in that case, it may have been a reverse genetic. But so yes, there was some overlap, but many of the genes that we got through GWAS were novel. So I think, in fact, that's perhaps not surprising. They, effectiveness is really the culmination of thousands of genetic interactions, and the genes that come through that are as crucial from genetic studies are perhaps so important that you can't really afford to lose them. So it's perhaps not surprising that we'll find novel, more subtle genes. Many of those probably wouldn't have a dramatic phenotype if you knock them out. I would expect some of them are redundant. So I'm keen to see whether we can use that genomic information, the association genetics, if you like, to try to predict which sets of genes or which haplotypes are optimal, and looking towards interactions with Ben Hayes and Lee Hickey and others in that area of genomic selection. Can we push the dial? One thing I didn't say is that there's apart from variation within Medicago, there's well-known variation in other species for nitrogen fixation effectiveness. and even major differences between species. So some, modern day varieties in soybean are quite effective, whereas in other species, they're known to, for instance, common bean, they're known to be relatively ineffective. So I think there's opportunities in various species to improve nitrogen fixation if we only focus on that as a trait. And until now, breeders have simply looked for yield and not necessarily for nitrogen fixation per se. So I think by focusing on traits like that and measuring them specifically rather than by virtue of yield as a sort of a general trait, I think we can move the dial on that.
Sure. Seeing no one else has asked me a question, I'll ask another one. In terms of the later stuff in your presentation about the future vision of tropical pulses, I'm just interested in what is the projection of the demand for these proteins and the domestic versus international demand. I mean, is it an industry that you see will be primarily servicing export, or is it sort of, is there anticipation that there'll be a strong development in domestic demand?
Well, I think the latter. I think there's an anticipation that there'll be a growing domestic demand. Most of our traditional importers or traditional clients, if you like, are in Southeast Asia, so they have cultural connections to whole foods. Whereas I think new markets, whether they're in Australia or in the developed world, that can afford these products will be new. So I think there's opportunities for growing export of manufactured foods, but I think most of these predictions are for a domestic market.
Yeah, right. Okay. Well, look, we'll wrap it up there. Can you just flick up Shahram's?
Oh yeah, sorry about that.
No, that's all right.
Here we go. I'm not going to see that unless I share my screen.
If we can.
Okay. Well, let's see. Is that's?
Oh, look, that's okay. Look.
Can you see it?
No, not yet. But we'll.
Why didn't that share? I'm going to shut. Well, keep talking. I'm going to.
Okay. Yeah. Well, first of all, I again want to thank Comms for helping with keeping the Coffee Science Seminar going when there's been a major change of staff in that group. So I just want to thank them. Obviously, I wanted to thank Michael for the great presentation and for your attending. Next week, we're going to have Shahram Nicholas from CNAFs, and he's going to talk about gut functionality, the key to sustainable poultry production. So again, I'm sure Shahram's a postdoc with Eugenie, and I'm sure that'll be a really valuable Coffee Science seminar as well. Now, these Coffee Science seminars will be also made available online through YouTube. So I'll close it up there and thank you all for attending. Okay, thanks, Michael.
Thank you, Craig.